Good day, class. I welcome you to another study session in biochemistry, which is study session 5, and we discuss the chemistry of nucleic acids. Nucleotides may be considered one of the most important metabolites of the cell. Nucleotides are found primarily as the monomeric unit comprising the major nucleic acids of the cell, which is ribonucleic acid, RNA, and deoxyribonucleic acid, DNA. However, they are also required for numerous other important functions within the cell. At the end of this study session, you should be able to define nucleosides and nucleotides. You should be able to explain the concept of nucleic acid. You should be able to explain deoxyribonucleic acid and ribonucleic acid. What is nucleosides and nucleotides? Nucleosides are glycosylamines that can be thought of as nucleotides without a phosphate group. It consists simply of a nucleobase and a 5-carbon sugar in a nucleoside. The base is bound to either ribose or deoxyribose via a beta-glycosidic linkage. Nucleosides include cytidine, uridine, adenosine, guanosine, thymidine, and inosine. Nucleotides are the molecular building blocks of DNA and RNA, and they are composed of a nucleobase known as the nitrogenous base. It contains a 5-carbon sugar, which can be ribose or deoxyribose, and one or more of phosphate groups. What are the components of a nucleotide? The components include purine, which are called adenine and guanine, and the derivatives of pyrimidine, which is single ring bases and are called thymine, cytosine, and uracil. On your screen, you can see the structure of the purine and the pyrimidine components of the nucleotides. Nucleic acids. Nucleic acid, like I've said earlier on, they are made up of DNA, RNA, and protein, and they are non-proteinaceous nitrogenous bases made up of monomeric units called nucleotides. They are composed of carbon, hydrogen, oxygen, nitrogen, and phosphorus. In 1935, Watkin and Crick were able to propose a double helix structure of DNA. Also, Crick suggested a term known as the central dogma of molecular genetics, which states that genetic information flows from DNA to RNA and from RNA to protein. Looking at the structure of a nucleic acid, nucleotides are joined together in DNA and RNA by phosphoester bonds between the phosphate component of one nucleotide and the sugar component of the next nucleotide. More and more nucleotides can be added on by the same process of forming ester bonds until an immense chain is formed. In the polynucleotide chain, one end of the nucleic acid molecule always has a free hydroxyl group on the sugar at the carbon known as the carbon 3' end, and on the other end of the molecule always has a phosphoric acid group at the 5' end. The counting system allows the strand of nucleic acid to be oriented. The 5' end of the molecule always ends with a phosphate group, and the 3' end of the strand always ends with a sugar. Deoxyribonucleic acid, which is DNA, is a very large molecule made up of a long chain of subunits called deoxyribonucleotides, attached to each other by phosphodiester linkages. Each deoxyribonucleotide is composed of deoxyribonucleoside and inorganic phosphate group. Each deoxyribonucleoside is composed of nitrogenous bases and a pentose sugar called deoxyribose. The nitrogenous bases are purines and pyrimidines. Purines are double rings comprising a 6-sided and a 5-sided ring. The two purine bases present in DNA are adenine and guanine. We have two types of DNA. We have the circular DNA and the non-circular DNA. What are the functions of DNA? DNA is used to store genetic information. DNA stores the code for protein, which exhibits traits. They get converted to RNA in order to move out of the cytoplasm. In the cytoplasm, DNA meets up with the ribosome where it can synthesize protein in the process of translation. DNA maintains growth and repair, and DNA controls all cellular activities in the body. Let's now move to ribonucleic acid, RNA. Is a biologically important type of molecule that consists of a long chain of nucleotide units called ribonucleotide. Ribonucleotide differs from deoxyribonucleotide in that ribonucleotide contains the O prime in the carbon 2 sugar ribose. Ribose is a sugar like glucose, but with only 5 carbon atoms in its molecule. RNA is found in the nucleus and cytoplasm, and it has linear single-strand of nucleotides. RNA contains ribose sugar and nitrogenous bases, the adenine, uracil, cytosine, and guanine. RNA backbone is of the ribose sugar and the phosphate group. Types of RNA. There are major 3 types of RNA. We have the messenger RNA. The messenger RNAs are the nucleic acid that record information from the DNA in the cell nucleus, and they carry it to the ribosome, and they are known as messenger RNA. We have the transfer RNA. Transfer RNA, the function of transfer RNA is to deliver amino acid one by one to protein chains growing at ribosomes. Then we have the ribosomal RNA, which exists outside the nucleus in the cytoplasm of a cell in structures called ribosomes. Thank you for listening. Have a good day.